Good evening everyone, and welcome if you're outside the University of Canterbury. Welcome here tonight. This is our What If Wednesday series. This is actually the first one I've ever been to, and it's a series that's held there's a talk every 2 nights. So just a couple of things before we start. First of all, please make sure your cell phones are turned off. Also, it's helpful if you can leave questions to the end because they can't be picked up on the recording. There will be time at the end for questions. And what else? Well, welcome, I guess. I'll just introduce the speaker. I'm Mike Steele. Hi.
Volume up, please.
Turn the volume up. Okay, I'll try that. Okay, a bit better?
Say something, and we'll know.
Right, so what I propose to do tonight is tell you a little bit about the way in which mathematics is used in the study of evolution, and in particular the inference of evolutionary trees. These are trees that describe the relationships between species and how they trace back to a common ancestor. So for example, this is a tree of mammals. So the talk's going to be really in 2 parts. The first is about why trees, and then why mathematics, and what are some of the problems that arise when you try to infer trees from data. In the second part, I'm going to show you some of the ways in which probability theory can be used to study these evolutionary processes. So the idea that there's some sort of hierarchical pattern or tree-like pattern to nature is a very old idea. The Greeks, of course, had the great chain of being, but I guess we almost daily refer to the Linnaean hierarchy when we give the genus and the species name, Homo sapien. So Linnaeus, a Swedish botanist, long ago realized that you have species and they fit within a larger group called a genus and so forth. And he constructed this hierarchical way of classifying nature. I've always had trouble trying to remember how this goes, and this German mathematician once told me, he said, "Mike, it's really very easy. It's King Philip comes over for green soup." So ever since then I've always been able to remember it. Well, this is just really some description of how to organise nature, but it was really Darwin who interpreted trees and hierarchies as an actual description of how evolution occurred, how species might have traced back to a common ancestor, and each of these branches in the tree would represent some sort of splitting event where maybe a new species would split off. Soon after the origin of species, a number of people started trying to produce these trees. Ernst Haeckel, in particular, constructed some glorious pictures of what we today might call the tree of life. And this tree has really 3 main branches. Over here we have the plants. On the right-hand side we have the animals. And in the middle we have everything else, basically bugs, things you might see through a microscope perhaps. And I think that's probably the way a lot of people think of life today. There's plants, animals, and there's everything else. But of course, the genetic story paints a very different picture. And when people started comparing the genetics of different species across the tree of life, they found that, in fact, the plants and the animals grouped together in a little corner of one of 3 main groups. So that's this group of eukaryotes. This is what you might think of as the bugs like bacteria, E. coli and so forth. But then there's this whole other group that is genetically different, and also actually if you look closely physiologically and cellularly different to bacteria as bacteria are to plants and animals. And they are the archaea. These are primitive single-celled organisms often living in extreme environments like hydrothermal vents or hot sulfur springs, and have a very different biochemistry, but also a very different genetic makeup. So this tree was constructed purely from genetic data. So you see the picture of life is very different over here. Plants and animals out in one little corner with fungi and everything else on the other two branches. And this gives us perhaps some hints as to where one might look for the earliest life, the origin of life. Well, since those days, biologists have got very good at building very big trees. This is a tree from the lab of David Hillis from Texas. 3,000 species, just mostly plants and animals, but there's a few other things down there. You can see in the animals that is a little in small writing, if you can read it, "You are here." Okay. So these trees are becoming very fashionable. In fact, you can even get them tattooed if you wish. I wouldn't recommend it because the biologists are always changing their view about how the tree should be arranged. Okay, well, trees like this, of course, are very hard to make sense of. They're just sort of a big mush. And so it's useful, therefore, to come up with ways of navigating through that tree. Actually, there's a mathematician, James Rossendale, at Imperial College. He's come up with a very nice program called OneZoom, which any of you can download for free off the web. And just for this talk, actually, he has kindly provided a little demo of how this program works. So here are the tetrapods, the things with 4 limbs. I asked him, could we fly in and see where humans are? And as we fly in, we'll pass through, of course, through the primates. We'll pass by things like, near the lizards and snakes. So just watch closely. Here we go. And as you go, we'll see also the times at which different lineages split off. Here we go. So we're coming into the primates now. And great apes, and there we are. Okay, so there's also some colour coding. The red is endangered or more rare. Of course, you see humans are not at all endangered in this picture. Okay, I don't know if you want to see that again. It's sort of perhaps worth watching twice. There's the tuatara out to the right there. Okay, so evolution really has 2 aspects. There's the idea that all life traces back to a common ancestor and the pattern of branching that leads to what we see today. That's mostly what I'm going to be talking about in this talk. However, a lot of the mathematics that's gone into the study of evolution has dealt with what we might call the process. That is, how is it that things change over time, the genetic composition of populations. And this is natural selection. So Darwin and Alfred Russel Wallace, who both in some ways independently came up with this idea of natural selection, unfortunately they didn't really understand in those days the mechanism of inheritance that Mendel later, well, actually was realised later Mendel had discovered, but it was rediscovered, those earlier findings. And then putting those two together, there was a lot of activity by theoreticians, mathematicians and statisticians, particularly in what became known as the new synthesis. So I'm just going to spend a couple of minutes talking about process, about the mathematical study of natural selection, just because it is a big part of the history of this topic, but it's not going to be much of the topic of my talk. So the 3 pioneers, or 3 of the pioneers I should say, were Ronald Fisher, who's well known to many of you who've studied statistics, and his American counterpart and sometimes adversary, Searle Wright, and this colourful character, J.B.S. Haldane, who used to come up with some delightful one-liners. The one I like particularly as a mathematician was an ounce of algebra is worth a ton of verbal argument. Okay, so these guys pretty much figured out how natural selection could lead to evolutionary progress, if you like. But there were some puzzles, things they couldn't really explain. Puzzles like, how do you explain animal behaviour? Why is it, for example, that some animals put themselves at a disadvantage to help others? That doesn't seem to be a sensible thing for natural selection to select for. Altruism, in other words. Also, why is the sex ratio so stable? These puzzles were really solved by a later generation. Bill Hamilton was perhaps known to you fairly well, almost a New Zealander. Both his parents were New Zealanders, but I think he was born in Cairo. And John Maynard Smith, who is, I guess, regarded as the father of what's known as evolutionary game theory, trying to explain animal behaviour and various other phenomena in biology. This guy on the left here is probably no one has heard of, but in his time, he died in 1975 in rather tragic circumstances in London. The only people that came to his funeral actually were these two guys and a whole lot of homeless people. Anyway, he discovered a remarkable theorem, very simple theorem, but nobody had noticed it. It had been sitting under everybody's noses, Fisher and everyone else. They'd completely missed it. And his theorem, in some ways, Price's theorem it's called, it's even used in economics today, it really makes sense of all of the earlier arguments that were taking place. You can understand and settle them very easily with that approach. So he'll be, no doubt, somebody whose name becomes a lot more familiar in years to come. Okay, that's all I want to say about process. I'd like to turn now to evolutionary trees. And this business of using genetic data or sequence data started, I guess, in the 60s with biologists looking at proteins, comparing them between different species and finding that when they built a tree, looking at comparisons between these proteins, they tended to agree fairly well with the fossil record. There were other studies by Cavalli Sforza, a linguist from Italy, and Anthony Edwards, a mathematician from Cambridge, looking at genetic markers in blood and producing trees. Mathematicians were also involved, two in particular, David Sankoff from Canada and Andreas Dress from Germany. But the whole business of building trees from genetic data was really driven by the data. What happened was there was a huge increase in the availability of sequence data. The cost of it came down enormously, and a large number of studies were then undertaken. So this is sort of the early history, I guess. This is just a little graph showing the number of papers published with the word molecular and phylogenetic both together somewhere in the abstract. So this finishes in 2000. This curve has continued to grow like this. So nothing much was happening in the 80s, but it's really taken off. If you look at the amount of data in genetic databases, a similar story. You had very little data there, and then suddenly it's this is 2002. This is like more than 10 years ago. This curve has continued to grow like this, but I just wanted to show you sort of the beginnings of this explosion in data. It's probably fair to say that the progress of genetic sequencing technology has way outpaced what you think of as impressive progress in computing technology. Okay? If you look at the cost of doing sequencing versus, say, the cost of doing calculations, it's considerably on the side of sequencing. Okay, what are these phylogenetic trees used for? Well, of course, they're used in systematic biology to describe relationships between species. They're used in conservation. You find some new organism, you don't know what it is, so you see where does it fit in the tree. They're used also in epidemiology, in understanding, for example, how different viruses like HIV or influenza, how they evolve. In this case, this is the Ebola epidemic from last year. You can take strains and reconstruct the history to try and perhaps find where did this start, how long ago, and so forth. In medicine also, we had a postdoc, Eric Matson, with us some years ago. He now runs a group in Seattle where they're using these techniques to study cancer. What they do is they look at the tumour and they regard that as really a tree of cells that started with the first mutated cells. So they're using those same techniques to study tumour evolution. And of course, those of you that have come to some of the Alan Wilson Centre talks will know the story of the famous work of Alan Wilson's group in Berkeley that led to the out of Africa hypothesis, the idea that all humans on Earth are descendants from a migration out of Africa very recently, 100 to 200,000 years ago. Very revolutionary idea when it came out in the 1980s, but one that is now generally accepted. So that work was very much reliant on phylogenetic techniques. But it's not just biology. Phylogenetics is used for classifying languages. For example, the Indo-European family. It's also, there's a field of study called stematology, where you take old manuscripts and you try and figure out which was copied from which, because in the old days before photocopiers they used to have a scribe who would copy and he'd make copying errors, so a bit like a mutation in biology. So you can use the same sort of techniques. It has even, I'm told, been used to classify single malt Scotch whisky. Actually read that paper. I was trying to see if I could get some grant money for that, but I think that one. Anyway, let's move on to why maths. Of course, we all know maths is fantastically effective in physics. It's almost too good, as Eugene Weiner, the Hungarian-American theoretical physicist, once said. But the situation with biology, well, it's like the poor cousin, I guess. This is what the Italian mathematician Giancarlo Rota once wrote. The real lack of contact between biology, mathematics and biology, it's either a tragedy, a scandal, or a challenge. It's hard to decide which. Okay, well, this was back in 1986. Maybe a few more things have happened since then. What about Darwin? Well, Darwin he studied maths, but he didn't really like it. He got a letter from his colleague Fox, who was having some problems at the time, and he replied, "I hope it arises from your being 10 fathoms deep in the mathematics, and if you are, God help you, for so am I. Only with this difference, I stick fast in the mud at the bottom, and there I shall remain." So he referred to mathematics as repugnant in one other place. Actually, we'll see towards the end of the talk he came round and actually wished he had, in fact, studied more mathematics. However, it's maybe, I've seen this view expressed, that maybe it was just as well he didn't know much mathematics, because with his knowledge, with the knowledge of the day of how inheritance worked, and this idea that the children are just some sort of blend of the parents, if he had tried to model natural selection this way, maybe he would have actually rejected the whole theory altogether. It was only really when the later reality of how inheritance works came along that natural selection can be put on a firm sort of mathematical footing. So why maths? Well, it's useful for analysing existing methods and developing new ones, but also for looking at a raft of other questions. Excuse me 1 second. Okay, so now we're going to do, there's very little maths in this talk. I think there's like 3 equations, but we have to have some sort of slightly formal aspect to it. So let's talk a little bit about trees. So we're going to deal mostly with rooted trees. The idea is you have at each point, this is the root, and these are the branches, and at each point you have a splitting. These are the leaves. This is the rooted tree. And this is actually, you're probably looking at that tree and thinking, nah, you haven't got that right. Why is mushroom with cat? Doesn't make sense, does it? Well, actually, biologists tell us it does. That is where mushroom goes, which is rather unsettling, maybe if you're a vegetarian, I don't know. Anyway, this is the rooted tree, but often we can think about what's called the unrooted tree, where we just really forget about where the root is. Now, why would we want to do that? There's several reasons. One of them is that the methods biologists use generally just give you an unrooted tree and then you have to figure out where the root should be. So the unrooted tree that corresponds to this would be this over here. So this tree, if I was to root it on this long branch here, if I was to, if you like, pull it down at that point, I would get that tree. Okay, so you have this sort of two ways of thinking about things. Right, so what's the first thing you do when you learn maths? You learn how to count. So let's count trees. And there's a proud tradition in mathematics of counting trees going back to the 19th century. Cayley's famous formula. We're not going to do that tonight. We're going to learn about a different formula for counting trees. This was discovered, very simple idea actually, but it is in the writings of mathematician Ernst Schroeder from 1870. So, of course, he wasn't interested in biology at all. It was just some mathematical exercise. 3 species. How many trees are there? Well, these species have to be the leaves of the tree. So we don't have too much choice. We've got one possible tree. What if a 4th species comes along? How many trees can I make now? Well, you see I have 3 branches. I should do it over here. 3 branches on which I can attach the bird. So I've got 3 ways. What if a 5th species comes along? Where can that go? Well, now you see I've got 5 branches. So how many trees would there be on all 5? On all 4 there was 3, and on all 5 there's going to be 3 times 5. So you see what's happening? What would be the next if another species come along? It would be 3 times 5 times 7, okay, because you're increasing the number of branches every time by 2. So if we had this number of species, just a handful, how many trees would there be? There'd be 10,395. Okay, well that's not too big. Any computer could look at that. What if you have 60 species? That would be a small study by today's biological standards. Well, the number of trees is just too big to even consider. Point of this is a really good reason for using mathematics, because you cannot simply look at each possible tree and say, well, how well does that describe my data? You need some clever way of quickly finding some sort of best tree. So, how do biologists go about building trees? In the old days they used fossils, or they looked at, do they have 4 legs or 6 or whatever? Of course these days they tend to use genetic data or genomic markers. And the basic idea is you compare different species according to characteristics they share but differ slightly on. So there's some traps though. It should be really easy, but of course it's not. Here's one trap. These 5 species, if I didn't know any biology, and some people might say I don't, what would the tree look like? Well, these kind of look similar, and these things are totally different. They fly, they've got wings, so the tree would probably be something like that. But that is not the tree at all. The tree is, in fact, like that. So what happened was you had something evolved to alligator and coming up here, and wings evolved up here but were lost down this lineage. They independently arose up the lineage on mammals, leading to the bats. So this is an example of what's called homoplasy. So we had a reversal, a character state was evolved and then undone, wings, no wings, and we also had convergent evolution. Okay? So homoplasy plays havoc with trying to build trees from simple comparisons like this. Now, when you look at genetic data, certain data has very nice properties that make it virtually homoplasy-free. And actually, there's a whole real interesting story on the mathematics of homoplasy-free characters, which I'm not going to talk about tonight. But an example of how this data is useful, these particular markers, signs, are called short and dispersed nuclear elements. How they're important is in doing things like resolving the mammal tree. The tree of mammals is pretty well understood, but there's some points that aren't totally pinned down. And one of the interesting applications of this marker was to figure out what is the closest living relative to whales. Whales, what do you think they might be? Well, it turned out to be the hippopotamus, and the strong evidence for that came from these particular markers. And when people then went back and looked at whales and hippopotami, they found there was actually a lot of reason to believe they were so closely related. Right, what are some of the other problems with genetic data? I'm going to talk about three briefly. One's called the Felsenstein zone, after Joseph Felsenstein, a statistician in Seattle. The idea is if you've got some sequence data, and what often happens is this might be a sequence in species 1 that codes for a certain gene, and in species 2 it's almost the same, but you've had some sort of change, some mutation that then got fixed in that population. So there's a G there and a T there. Now these sequences are very, very long, so you have these, every now and then you have a difference. And so you have some sort of model of change between these four bases. Now if this was the true evolutionary tree that described the history of these four species, then as an unrooted tree it would look like this, 1 and 2 and then 3 and 4, okay? But suppose that the rate of evolution, the rate of change between these bases on this lineage here leading up to 2 was really, really high, then what's going to happen is that species 1 and 3 will nearly always, if they're close together in the tree, they'll nearly always be in the same state because there's been very little time for there to have been a change. Whereas 2 will be, maybe it's changed several times, so you could think of it as almost as random, and so will 4 because it's very far away. So just by chance alone, you're going to get a lot of patterns that will put species 1 and 3 in a single state like A, and 2 and 4 in a different state like C. And that's going to favour this tree, 1, 3 versus 2, 4. And that's exactly what biologists find happens with certain data. It's called the Felsenstein zone. Maths is useful for two ways. Firstly, to say how often would this happen, what are the conditions on which it would occur. But more importantly for the biologists, it provides a way of correcting for this inconsistency. One can formulate a new method based on maximum likelihood that is able to consistently recover the correct tree. So given enough data, it will be sure to find the correct tree under that model. A similar sort of story, this was a study we were involved in some time ago now. The fossil record tells us that birds and crocodiles are sister taxa when you compare them with mammals. But when biologists started looking at a certain type of data called 18S ribosomal RNA, it was always, for some reason, putting mammals with birds and crocodiles out there. And just any method you use seemed to just tell the same story. Very, very odd. Why was this? Well, it turns out that independently in both of these lineages, to birds and to mammals, their sequences were becoming rich in G's and C's. So again, just by chance alone, you're going to get a lot of patterns that will put those together. So once again, you can construct a method that will correct for that and recover a consistent evolutionary story. Okay, so that was the first problem, Felsenstein zone. The next one is called incomplete lineage sorting, and it illustrates something which is the difference between a species tree and a gene tree. So suppose we take these three species, human, chimpanzee and ape. So when I was doing my PhD, biologists were still debating vigorously what was the relationship between these three. It had been assumed that probably chimp and gorilla were more closely related and human had come off earlier, but most of the genetic genes that were being studied seem to often show this relationship. Now, a lot more is known now. We have a lot more genes, and this is now the accepted species tree, that human and chimp are sister taxa with gorilla coming off earlier. Nevertheless, some genes tell a different story. Why is that? Well, if I take a human, a chimp, and a gorilla, and I sequence this gene from them, now Mick got that gene from either his mother or his father, who got it from their mother or father, and you trace this back. Trace back the history of that gene going back in time. Eventually, these two copies will coalesce, and then the one copy from the gorilla will coalesce. In that case, the gene tree tells the same story as the species tree. It puts human and chimp together and gorilla as an outgroup. But it could be that when we do this, these don't coalesce in here, they come back, and in that case, we get a different gene tree, one that puts gorilla and chimp together. The good news is, though, that this doesn't happen very often. You can prove, in fact, that the most likely gene tree will always be the same as the species tree under simple assumptions, and that the support for the other two trees will be roughly equal, and this is exactly what people find in studies. So that seems like, well, that problem's solved then. You just sequence lots of genes and take the majority vote for what the tree is. Well, that works for 3 species, but unfortunately it doesn't for more. This guy, James Degnan, was actually a lecturer here until recently, came out from America. He and Noah Rosenberg discovered something rather startling, that whenever you have 5 or more species, the most likely gene tree can be different from the species tree. It was a big surprise in the systematic biology community. So any voting strategy is not going to work. You need to be more clever. And since then, a number of bright people have come along and really mathematicians, mostly probability theorists, they've come along and really solved this problem. How do you combine gene trees into a species tree in a consistent way? So actually all 3 of these are from the United States. Okay, the third problem with building trees is when you go way, way back in time, this is like maybe 550 million years ago, the origin of animals, but you go back, say, to the origin of photosynthesis much, much earlier, maybe 1.5 billion years ago, or closer to the origin of life, the signal that's in DNA gets sort of swamped, if you like, by random effects. And a nice way to display this is by a method that's become very widely used. It was invented by 2 mathematicians. One of them, David Bryant, was a PhD student at University of Canterbury, and this method sort of shows you the conflicting signals for different trees. So this is a really interesting story, but I don't have time to go into it right now. Okay, so that's part 1 of the talk. Part 2 is on probability models. And this has quite an old history in a way. George Houdini-Ewell was sent some data by one of his colleagues in the Royal Society, and he was interested in this question, why is it that this is the number of species in a genus. So in this study, this is of snakes, there's 131 genera that just have a single species in it. Okay, and then there's 35 that have 2 species. So lots of genera with just 1 or 2 species, but then you have this long tail. And he asked, this is Willis, I think, Dr. J.C. Willis asked George Houdini, he sent him lots of data sets, not just this, can you come up with some sort of explanation for this pattern I'm seeing? And Ewell had this idea, well, let's just consider a simple model where new species form at some random rate, and every now and then there's an event where a new genus is established, a new species that's so different from everything else. And so this model really just has 2 parameters, the rate of speciation, the rate of genus formation. If you take their ratio, Ewell was able to come up with this very nice formula for the probability distribution for the number of species per genera, and it fitted his these observations pretty well. So we're going to use Yule-type models here in what follows. But first I want to take a step back and say, where do these evolutionary trees come from? So I kind of think as a mathematician, the biologist goes out into the field and saws down his tree and brings it back proudly and puts it on the table for me to admire. And that's the evolutionary tree. But that's not really where they come from, is it? A much less anthropocentric viewpoint is this, that actually evolution has been happening through time, species have been splitting off, some going extinct. We just happen to have come along at the present and looked across and said, I see some things there, so here's my tree here. Okay, so it's sort of like cutting a tree off, not at the bottom, but near the top. So what does this difference in perspective mean? Actually, we'll see it has an important influence. The question I'm going to be interested in is how long are the branches in a tree? So we're going to assume that speciation occurs at a constant rate. Every second there's a chance there'll be a speciation event. Technically it's called an exponential distribution, but it's just the same distribution as you'd use as the waiting time until some random event happens if those events occurring at a constant rate. So I'm going to assume there's a speciation rate of 1 in a million years. So the time from here till the next speciation event, on average, it'll be 1 million years. Okay? So, but on the other hand, I've just looked across. I haven't turned up at the end. I've just looked across here, and so now I want to know what's the time here. Now here we have to be a bit careful because this is something actually quite useful to know. I do this with my first year stats class. You turn up at a bus stop with no idea when the next bus will arrive. If the buses arrive regularly every 20 minutes, what is your expected waiting time? Well, 20 minutes, 20 minutes, 20 minutes. You arrive anywhere in between. On average, it'll be 10 minutes, okay? Not rocket science. But if the buses arrive randomly every 20 minutes, on average one every 20 minutes, what's your expected waiting time? Is it the same? Is it better to live in a world where they arrive randomly or regularly? Well, your expected waiting time, if this is like an exponential again, here it was 10 minutes, here it's actually 20 minutes. So why is that? It's because you're more likely to arrive in a longer gap between buses. So we have to bear that in mind when we think about these questions. So there's some puzzles in this talk. So a tree evolves with each lineage randomly generating a new lineage on average once every 1 million years. Let's forget about extinction for the time being. Let's look at the tree when it's got 100 leaves. What is the length of a randomly selected extant branch? What's an extant branch? It's a branch that's still live. So this is where we've looked at it here. It's got 100 leaves. If I choose one of those, what's the expected length of it? Well, so these are the interior branches. So for the extant branches, is it 1 million years or 500,000 years? You kind of think, well, I've just turned up and sawn some of those pendant edges off. It's going to be less than 1 million. It's probably 500,000, right? But then you think of the bus paradox, and you think, well, it's like waiting for the bus to arrive, so maybe it's 1 million years. But it's not that either. It is actually 500,000 years. It's exactly 500,000 years, in fact. It's a little surprising because the software biologists were using for dating these trees was assuming it was in fact 1 million years. Okay, what about these interior branches? Now those interior branches you haven't touched. So they surely be 1 million years. No, they're still 500,000 years. Very surprising. Anyway, I can talk about it later if you like. Okay, now suppose that extinction occurs at the same rate as speciation. Reconstructed tree we see today as 100 tips. So would we expect this tree to just look the same as a tree that was evolved without? The reconstructed tree is just the tree when we look across at the present and we forget about all these things that have gone extinct. Would it look the same as just a tree that was growing without extinction but at a lower speciation rate? Turns out no. trees with no extinction look like this. Trees with extinction equal to speciation look very, very sort of thin at the top, very, very short branches at the top and big long ones down near the bottom. Now it turns out that real biological trees look way more like this than like this, which is a bit of a paradox because we all know speciation is almost as prevalent as extinction. But what's going on here is simply the fact that if the extinction rate was actually equal to the speciation rate, the tree is guaranteed to die out. It must die out. It's a simple fact of probability theory. So as soon as you make the extinction rate just a little bit less than the speciation rate, you return very quickly to these sorts of tree shapes. So why is this relevant? Well, this is a question that we got interested in a few years ago. Suppose a random 10% of species from some clade were to disappear in the next 100 years due to current high rates of extinction. How much evolutionary diversity would be lost? Okay, well, this is a tricky question, as we know. It's difficult to predict, especially if it involves the future. This was the tree of birds, for example. There's 10,000 species of birds. In building this tree, they showed exactly how much evolutionary history is endangered. So these 575 species at the tips represent 2.7 billion years of evolution. What is this evolutionary heritage measuring? So in the old days, conservation, you'd say, well, how many species will be lost? But nowadays, people are saying, well, if for example, a tuatara goes extinct, that's 200 million years of evolution back to the nearest thing. It's a very unique and very different species. We shouldn't just count it as a single species. So that's on equal weight with everything else. So this idea of evolutionary heritage, if you have a tree, suppose these three species survive, the remaining heritage is just the sum of these branch lengths. So you're missing out on this length and this length. On the other hand, if those two went extinct, you'd also lose that earlier branch length. Right, so let's suppose that let's try and predict the proportion of diversity that remains if each species survives with a certain probability. And the reason we were interested in this is what was done in this paper by Ni and May. And they made this rather provocative claim that 80% of the underlying tree can survive even when approximately 95% of species are lost. A claim that has been, this paper has been cited many times, well over 200 times, and it seemed a bit surprising, this finding. And when we looked at the methods they used, they were really using this model of tree shapes, which are kind of not really the trees you see in biology. So we thought, well, let's use these Yule pure birth trees, and let's do the following experiment. We're going to grow such a tree, and then we're going to prune some of the leaves at random. So notice there's two random processes going on here. There's the random tree and then randomly killing off some leaves. And we're going to look at what's the diversity of what's left, the sum of the branch lengths, divided by the total sum of the branch lengths of the tree we started with. So it's a ratio of two random variables. We thought this would be really hard to calculate anything about the mean or whatever expected value. Turned out to be extremely elegant in the sense that all the things that would seem to matter, like the number of species, the rate of speciation in time, disappear. And so here's our first equation. Future diversity divided by present diversity is just given by this very cool formula that only involves the proportion of species that survive. How it relates to this earlier paper is that instead of 80% of the underlying tree surviving, when 95% of the species are lost, we find that 84% of the underlying tree is lost when 95% of the species are lost. So you see that your choice of model can make a huge difference in your prediction. Finally, this will probably be finally, can we explain tree balance? You see, some trees that biologists produced are very unbalanced, like this tree here. If I stand here, I see one species on one side and a whole bunch on the other. Same if I'm here. This tree, on the other hand, is very balanced. Wherever I look, there's roughly half on either side. So the motto, less is more, comes into play here. You could imagine a very complicated model of speciation where maybe the rate of speciation depends on time. Maybe there's rapid bursts and long quiet periods. Maybe it depends on how many other species are around. And maybe the rate of extinction might even depend on where you are in the tree. But as long as whenever there is a speciation event, all of the taxa at that point are equally likely to speciate. When you look at the reconstructed tree and ignore the branch lengths, the tree shape always has the same probability distribution. So you have this remarkable robustness to the intricacies of the model in that way. This was David Aldous from Berkeley gave a talk out here a couple of years ago, and Tanya Stadler was also a PhD student here. So how do we calculate this probability? And this is where we're going to do a practical demonstration shortly. It's very simple. We start off with two species. And then what happens is one of them speciates. So maybe the left-hand one does or the right-hand one does. So we get one of those two trees. And then we've got one of these three species speciate. So we could get, if this leaf speciates, we get that tree, this one, this one. If this one does, we get a tree that's slightly different. We've got two leaves on one side, two on the other, and so forth. And this process grows. Okay, well, here's a question. It's called the Yule-Harding model. Let's grow a tree until it has 100 leaves. Which is more likely? A tree where you have 99 species on one side and one on the other, or 60 on one side and 40 on the other? Looks a pretty hard question, doesn't it? Well, let's see if we can solve this question. What's the probability of 99 in 1? Well, it turns out there's a really nice way of doing this by realizing that this is nothing more than what's called a classic polyurn. And so I just need to explain polyurn for a second. Polyurn is, there's no tricks here, there's nothing hidden. I'm not dynamo. So we have an orange ball and a white ball. I'm going to put them in the basket, and what happens is I pull one out and I look at it, it's orange. And I put it back in along with another one of the same colour. So I put another orange one in. And I just repeat this process. I just pull, of course I mix them around and I pull it out. It's orange. So I put another orange one in. Okay, and I keep doing this. Right? So this is the classic polyurn model. And we want to know what's the probability of a certain number of white and orange after a different period of time. Now, how does that relate to this problem here? Well, if you think about it, if you think, for example, that where I start with the two species, the orange one is the one on the right, the white one is the one on the, the one on the left, the white one's the one on the right. To follow that left-hand path up there, so we've got orange here, I need to pull out an orange one, then I need to pull out another orange one. So I'm really asking, what is the probability that when I do this process, starting with one orange and one white, I'm always only ever going to have one white at the end. So I'm just going to have orange and orange and orange and orange. What's the probability of that? Well, the probability I make that first step is a half. What's the probability I make the next step? So I either have to go there or there. There's three possibilities, but two of those three possibilities would give me three on one side and one on the other. Okay? So it's 1/2 times 2/3. And what will be the probability for the next time? So now I've got 3 orange ones and 1 white one. What's the probability I pull out an orange one? It's 3/4, and then I put another. Okay, so it's 1/2 times 2/3 times 3/4. What's going on here? What is this? And don't rush for your calculator. What is this? Well, 2 cancels the 2, 3 cancels the 3. 1/99. There, done without any calculation at all. We can calculate the probability of having this tree here. Now what about this tree? This is going to be way more complicated, right? But the wonderful thing about the Polyurban model is that this will be exactly the same probability as that. It's quite easy to show. So what could have been a disastrously complicated calculation with a little bit of classic maths, this is a well-known result from long ago, from the 40s or 50s, the fact this is a uniform distribution, you can calculate, you can answer that question now without even getting out a piece of paper. Yeah, that's how mathematics should be done. So how well does the Yule-Harding model describe the shape of the trees that biologists build? Quite well. For some reason, real phylogenetic trees tend to be slightly less balanced than the Yule-Harding model predicts. And the question of why is a really interesting one that a lot of people, like Tanya Stadler and some of us, have been working on. No, we're not going to do that. We're not going to do that. The last common ancestor, we're going to go straight to the end to talk about Darwin's regret, because we've run out of time. So give another talk sometime about the last common ancestor. Yeah, I said that Darwin really didn't like maths, and maybe it's just as well he didn't. But later in his life, when he was writing his autobiography, he talked about how he attempted it and had a private tutor, a very dull man, he says, to Barneth, but I got on very slowly. But then he says, this impatience was very foolish, and in later years I've deeply regretted that I did not proceed far enough, at least to understand something of the great leading principles of mathematics. So there we are. So enroll in your maths course and come to Canterbury. Okay, thanks.